questions, by now you should know me. Today I'm going to be walking you through Keldol and what you're going to be doing before lab as well as what you will be doing during the lab. So before lab, I'm going to prepare all the steps through digestion for you, and then during lab we'll be doing the distillation. The first step that we're going to do that I'm going to show you right now is weighing out your samples. And the way that we do that is we are going to take nitrogen-free weigh paper and we are going to fold it up and tear it out on our scale. So then we know, based on the estimated amount of protein in our food sample, how much we need to weigh out. So for almonds, which I'm weighing out right now, we need to weigh out 0.25 grams. So once we tear out our weigh paper, I'm going to gently weigh out the 0.25 grams into the center of my paper to make sure that I don't lose any of my sample. So the reason that we are using nitrogen-free weigh paper is because the whole weigh paper with our sample is going to be transferred into our digestion tube and digested together. So if there was nitrogen in our weigh paper, then we would overestimate our protein concentration or content from what is in our sample. So I went a little bit over, but that's okay because I'm going to record the exact weight of our sample. So then I'm going to take out my sample from the scale. I'm going to put all of my sample into the middle of the weigh paper and fold it up nice and tightly so that none of it falls out or gets blown away as we are transferring our sample into the digestion flask. And now our sample is nice and secure in there and it's ready to go onto the digestion. Hi, okay, so now that we've weighed out all of our samples, we're going to the digestion step. So this is our digestion unit, it's a Buki unit, and here what we're looking at is a hot plate with a series of dips in it that fit in our digestion flasks where we will put in our sample along with the reagents for the digestion step. So all of our flasks in the rack are labeled, and we're going to put in specifically which rep into each of these and make sure that the sample is in there, there's nothing flying up here, and then the solution that we have for digestion covers our sample for complete digestion. So for now, before we start the heating block, we're going to add our samples into each of the digestion tubes, and then we're also going to add a piece of nitrogen-free weigh paper into the digestion tubes that are for our blanks so that we can get a measurement of the weigh paper apart from the sample to subtract later on in our calculation. We're using nitrogen-free weigh paper for our blanks because if we had nitrogen in there, when we went to go measure the Keldol, because the measurement for protein is based on nitrogen, we would be overestimating our protein content in our food. So that's why it's important to take a blank measurement and also that it's a nitrogen-free transfer into our tube. So for each of your samples, we have folded them up really nicely and we are putting them into the tubes with the weigh paper so that we can ensure they drop all the way to the bottom and no sample gets on the sides of our tubes, because if it's on the sides, then we won't be able to digest it here at the bottom for a complete digestion. Now, my trusty assistant, Leslie, is going to add in 2 Keldol tablets. And what these are is that they're a catalyst. So they speed up the digestion reaction. And the way that they do this is that they're made of copper sulfate. So the salt content in them helps to increase the boiling point of our sulfuric acid that we're going to add also to these tubes, so that we can increase the temperature at which we are doing the digestion step on our digestion block and make the reaction go a little faster. By increasing the boiling point, the reason we're doing that rather than just increasing the temperature is because we're going to put this fume hood onto our samples, and it's going to create a vacuum in which it will pull off any of the fumes that are coming off. But if all of our acid is going to be boiling, it would take all of our acid out with the fumes, and then we wouldn't have a digestion within our tubes. So now we've added our sample, the Kildall tablets, and the concentrated sulfuric acid to our digestion flask, and we can see that it's starting to turn brown, or it's starting to digest everything in our sample, such as our fats, all of the organic matter. So once it starts to do that, it will actually turn our sample black, and then when we heat it and it completes digestion, you'll see our sample actually turns blue from the copper that's present in our catalyst, the copper sulfate. And that's how we would know that digestion is done. If we had any sort of black particles in there, we would be able to tell that digestion was not completed and we would need to do it further. So we're just making sure that all of our sample is in the acid. And now we are going to start warming up our heat block so that we can start the digestion. So now that the heating block is heated up and it says that it's ready, we're now going to lower our digestion tubes onto the heating block and start our digestion step. So it will now heat at 420 degrees Celsius for 2 hours. So it's important that we keep this fume exhaust on, and we are going to hit start, and now it will count down for 120 minutes. So the fume exhaust, whenever we're going through the digestion, it will start to release acidic fumes. And then those fumes will go through the condenser here and then be neutralized here in this sodium hydroxide solution. Here we can see that the solution is blue, and it's supposed to be replaced when it's yellow or green. And that's because the indicator means that it is alkaline, and once it gets yellow or orange, we know it's acidic and needs to be changed so that we can further neutralize any fumes moving forward. So now we can see that the digestion has started, and those fumes that are coming off are being sucked into the manifold through the exhaust, and then they are coming through and being condensed with cold water here, and the bubbler is going so that all of the condensed fumes are going through the sodium hydroxide solution to neutralize. So now it has been digested, all of our samples, and we can see that the samples are no longer black, but they are kind of neon green or blue in color, and that's from the copper that was in the catalyst. And so we've now pulled up our samples to cool while keeping the exhaust on to take out any extra fumes that are coming off while cooling into our bubbler. And so we can see into the bubbler or the scrubber that the color on here, if Brigida could get the side of it, has changed from that dark blue color now to the yellow color. So our acidic fumes are now in the bubbler, and we'll need to replace this for the next digestion that we do. But for now, we are going to, when cooled, add water, about 50 milliliters of water, into each of the samples, and that is to make sure that they don't crystallize. And then your samples, when they are cooled, will be ready for the distillation and titration, which we will be doing in class. Okay, so now that we've finished the digestion and it's cooled down, I've added DDW, or double distilled water, into each of the flasks to dilute our sulfuric acid, our digested sample. And we can see that the color is now a light blue color, and there is no black in any of these that would suggest that we have any undigested particles. So now what I'm going to do is I'm going to take off the manifold, and we are going to start with our titration, distillation, neutralization, and then titration of our samples. So each sample you have will have a sample blank, and then your 2 samples that you're going to run. I'm going to start with my sample blank, and we're going to take it over here to the distillation unit, and we are going to carefully remove the last sample and put it into the rack, and then we're going to place our blank sample onto the distillation unit and clamp it in there tightly to make sure that there's a tight seal around that O-ring, because as it starts to distill, you don't want any of it to leak out. Now we need to prepare a flask to distill in. So you'll come over here and you have all of your flasks ready for you, and you can see all of your flasks correspond to a specific rep. So each repetition has a corresponding weight to it, and you'll need to keep those straight so that we can know in your Excel sheet exactly how much nitrogen is in how much sample. So because I'm doing my blank, I'm going to take my blank flask and I'm going to add 50 milliliters of 4% boric acid to it. And there are transfer pipettes here if you need to use them. And then I'll add 50 milliliters to also my sample that I'm going to be doing. Now to each of these in our addendum, we can see that it says you're going to add 2 to 3 drops of methyl red indicator. So here we have our methyl red indicator. I'm just going to take a transfer pipette and add 2 to 3 drops to each of these. So this is the color that you want your samples to be when starting out. When we start the titration, you'll see that they start to change colors, and that's due to a change in pH. So now we're going to take our blank flask and bring it over. We're going to remove the holder flask, set it aside, and we are going to add in our blank flask. So now what I'm going to do is I'm going to do the neutralization. So in our sample right now, we have ammonium sulfate. And when we add the neutralization, or our 32% NaOH, what it's going to do is release that ammonium into ammonia so that we can distill it into our flask. So as it heats up, the ammonia will come through here, and we have a condenser here that is run by cold water. And the ammonia will condense down into our boric acid, and then that creates a borate ion. And so that's what we will use for the titration part. So here, because we have 30 milliliters of concentrated sulfuric acid that we added in the digestion, we can assume if we have 32% NaOH that we need to add at least 3 times the amount of solution or NaOH to our sample flask. So I can see on here that there's a graduation at the back that tells us exactly, maybe I can open it to give us a better look, tells us where we are on our flask. So I'm going to add 3 times the amount, which would be roughly 90 milliliters of NaOH, by holding down the reagent button until I get to that point. Okay, so now I have added 90 milliliters to our flask. So something that is cool about our system is that if you hold down the reagent button and dispense as much as you need for the first time, then it will have a memory of that for your next sample, and you can just hit it 1 time and it will dispense the same amount of reagent for each sample. So now we're going to distill it for 5 minutes, and we're going to hit start. So now steam will start to go into our sample and heat it up, and then the ammonium will start to distill down and be condensed and go into our sample flask. Okay, so now that the distillation is done, we can see that we have distilled some of our solution into the sample flask, for in this case the blank. So because our blank shouldn't have any nitrogen in it, we don't see a very big color difference. So what we're going to do is take our sample blank and tap it off, and then we're going to take our flask over and do the titration. So I like to have my undistilled solution here as a reference point, because we want to just get this blank back to where our reference point is for the titration. To prepare your titration, you are going to want to fill your burette. So you're going to fill your burette up to 0, and remember that it's important not to have any bubbles in your burette, or that counts for volume, and it can throw off your samples. So now for our blank, we are just going to get the blank back to our original color, which shouldn't take very much. So I'm just going to put in very slowly, a drop at a time, and make sure you don't have any drops that are lingering there that could fall, because that will count as volume also. So it looks like we need maybe another drop. And we are back at our original color. So that was just 0.2 of a milliliter, as I can see from the meniscus on my burette, and I'm going to record that titration volume. So now we're going to do our samples, and we're going to open the distillation unit, and you're going to use a hot hand because we use steam to heat this up. It's going to be very hot. So you're going to grab your flask with your hot hand, and you're going to open this and then slowly release it down and tap off the stem. Then you will put this over onto the rack for it to cool, and then your TA will take care of the waste from there. So now we can replace it with our sample flask. And here we have our sample flask, and I'm going to again install it the way I did before, push it up, snug against the O-ring, and then secure it into place. And I'm going to take my sample flask that corresponds to the correct one, so this is rep 1, and this is my first rep, and I'm going to place it underneath, and we will do the distillation again with the adding of reagents, and then 5 minutes to distill. And we will hit start. So now we can see that during the titration, or the distillation, that our sample is now turning yellow in color as that ammonia is being distilled into our sample. It is changing the pH, and the indicator is now more alkaline and moving to a yellow color. So now our sample is done. We can see that it has changed to a yellow color, and we can again take this and tap off the stem to get that excess. And then we are going to go titrate this. So again, as I'm titrating my sample, I want to get the color to return back to the original color. So I like to have my previous sample here so that I can compare the colors and make sure that we're titrating to the same endpoint and not overshooting it or undershooting it. So I'm going to slowly start with just dripping the HCl into my solution and swirling at the same time. You want to make sure to be careful not to get any of the drops onto the side of your flask, as that will count for volume, but it might not get down into your solution to actually titrate it. We're starting to get some pink color. And I'm going to stop it, swirl really, really good, and compare my colors. So I think we could call that the end point. And I'm going to measure. Again, we started at 0, and now I am at 10.9 milliliters of titrated volume. So we're going to write that down, and then once you finish titrating all of your samples, you'll have 2 sample reps and then 1 blank, and we've prepared extra in case anything goes wrong. But once you have your data, you'll come over to the computer, and in this Excel sheet, the TA will have put all of the sample weights here for you, and you have your product and then the replicates, and then it asks you for your volume of HCl that you've titrated. So we are going to put in here 10.9, as that was the first rep of the Parmesan cheese that I had shown you as an example. and you'll record that data. And then down here, you can see for the blanks, we have it by group. So I'm just going to pretend like I am group A, and you can see this is grayed out. We don't need a sample weight for our blanks, and I'm going to enter 0.2 as that's how many milliliters I had to put in for the blank. So then you'll do a subtraction of your blank value from your volume that you titrated for your samples, and then you'll be able to use your calculation that is in your addendum to determine the percent nitrogen. And here we have your nitrogen conversion factor listed for you. So we can see that there's different factors for each food type based on what they are. So for cheeses and most dairy products, 6.38 is typical. And then for other products such as baby cereal or almonds, it's lower, around 5.17, 5.18. So then you'll multiply your percent nitrogen by your factor to get your percent protein, and here you can record your averages, your standard deviation, your CV, your percent relative error, and then your dry basis, and then an average for your dry basis. And that's it.
Hi everyone, I'm one of your TA. My name is Fan, and today I'm going to talk about the Umass method we use to measure the nitrogen content of your sample and then eventually convert it to the protein content. So the Umass methods qualify nitrogen by combustion of the sample, and samples are weighted and combusted at high temperature. In this case, we have our instrument is 950 Celsius, which will release your nitrogen and other combustion products such as carbon dioxide, water, and so the combustion products are removed through various absorbents, and the nitrogen gas release is qualified using a thermal conductivity detector. Nitrogen content is determined using a calibration curve and then converted to percentage protein using a conversion factor that is based on the ratio of protein to nitrogen for that particular type of food. Before analysis, you need to turn on your gas tank, which is on the left. We have our numeric gas, which is the compressed air, and then we have our inert gas, which is helium in this case, and then we have our oxygen. So we will turn on those three gas tank, and then the second thing we need to do is turn on the gas bottom on the screen. And the third thing we want to do is to perform a system check. The system check will allow you to, the instrument will check every part of the instrument itself and to make sure everything is worked correctly, such as the temperature is within the range, and also the pressure of each part is within the range. And the fourth things we want to do is to perform a leak check. The function of leak check is to check each part of the instrument does not have a leak. Okay. After you make sure each part of your instrument, which is leak hole, is working well, you need the first thing you want to do is to run a bunch of blanks. So the reason we want to run a bunch of blanks is because the blanks actually measure how much air you introduce into the instrument. So the instrument will give you the end nitrogen content of your sample by subtract the amount of nitrogen in the air. So after you run a bunch of blanks, you need to run drips, a standard materials to make sure the instrument is working and reading correctly. So in this case, we have two types of standards we used. The first thing is EDTA. EDTA is used for when you're doing dry samples. So and we have glycine. We will dissolve glycine into solution to prepare standard solutions for liquid samples. Since in the class we only have dry powders and cheddar cheese and other cheese sample, we only run dry samples. So today I'm only going to correct using EDTA, plus we're using these caps. Those caps are designed for dry samples, and those tin capsules are designed for liquid samples. So when you're doing liquid sample, you're using glycine standard solution and the glass tin capsules. When you're doing dry sample, you want to use the EDTA plus the gel capsules. The reason we have tin capsules is because the gel capsules cannot contain liquid sample because the water will dissolve this cap. So that's why we have two different setups. These are 4 drifts I weigh out in the gel capsules. As a good student, you will also record the weight of the EDTA you weigh out. So the instrument gives you a range you need to weigh out. It's around 0.25. So make sure you need to weigh out in the range. You will enter the mass of your drift into the instrument. So here is the mass of my 4 drifts. And then the instrument will tell you to put the first of your draft into location 9. That means which is here. And then you will put another one into 10, 11, and 12. Okay. And then I will click the bottom instrument to analysis. So the sample dropped into the combustion tube. The only thing you will do in the lab is weigh out your samples into the gel capsules. So for cheese group, you need to first grind your cheese before putting into the gel capsules. So the first thing you want to do is grab a gel capsule and put into this and fill it. And use the tweezer to put the cheese sample into it. The important thing is you don't want your cheese sample to be outside your capsules, so you need to wipe them to make sure it's clean. And then you need to record the weight of your sample after you're done with weighing.